Welcome to this channel. In this video, we talk about transport of substances through cell membranes. This chapter will include active transport, diffusion, facilitated diffusion. So as we know, active transport, when molecules are moved against concentration gradient with expenditure of energy, it is active transport. So energy is required. While in diffusion, movement of molecules from an area of high concentration to an area of low concentration occurs and no energy is thus required. While in facilitated diffusion, which is also known as carrier-mediated diffusion, substances being transported by diffusion through a membrane using a specific carrier proteins occur. But since in facilitated diffusion, just like diffusion, the movement across the cell membranes happens from high concentration to the low concentration, that is along its concentration gradient, so no extra energy is required. Now in here, if we talk about the lipid bilayer, before we start talking about how transport occurs through it, we can see the fluid mosaic model here. The fluid mosaic model. In this model, we can see these are the lipid molecules, right, in two layers. These are the hydrophobic tails, while these are hydrophilic heads, right? So now we can see also that there are many proteins. All of these are membrane bound proteins, right? Some of these proteins are intrinsic, which move from this side to that side without anything. Now essentially all cell membranes are made up of lipid bilayer along with large number of protein molecules embedded in it. As we can see, the lipid bilayer is not miscible either with the extracellular or intracellular fluid compartments. Therefore, it constitutes a barrier against movement of water and water soluble substances, right? So lipid, as we know, are oily substances which is immiscible in water. So if this is extracellular compartment and this is intracellular compartment, the internal part of the cell, we can see this, there's water outside and there would be water inside. But because these are oily substances and we know that oils make a layer between water or above water, so this lipid makes a layer between extracellular and intracellular environment and thus maintains the two compartments individually. Now only lipid soluble substances can easily pass across the membrane by diffusion. The membrane proteins have entirely different properties for transporting substances, providing an alternative pathway of crossing the membrane. There are many types of carrier proteins or channel protein. Now the protein molecules of the membrane have entirely different properties for transporting substances. Their molecular structure interrupts the continuity of the lipid bilayer, constituting an alternative pathway through the cell membrane. Many of these penetrating proteins can function as transport proteins. Different properties function differently. Some proteins have watery spaces all the way through the molecule and allows free movement of water as well as selected ions or molecules. Now these proteins are known as channel proteins. Other proteins called carrier proteins bind with molecules or ions that are to be transported. Conformational change in the protein molecules then move the substances through the interstitials of the protein to other side of the membrane. Right, so here we can see this is a channel protein as it has been written here. This is a channel protein and it provides empty pathway through which the molecules in the extracellular compartment can move straight through to the inside of the cell. Well, there are certain carrier proteins, like if I can show you here, this is a carrier protein, if this is lipid bilayer. So the molecule will bind here, and after the molecule will bind, it will change its conformation will change, and then it will release the molecule on this side. So it does not have a channel or a pore through which the molecules can move inside, but it is a protein that changes its conformation to transport substances. This is carrier protein. Now if we talk about diffusion versus active transport and the difference. Transport through cell membrane occur Transport through cell membranes occur either via active transport or by diffusion, also known as passive transport, right? So if it happens with the gradient potential, that is, it moves across the membrane along with its concentration gradient from high concentration to low concentration, then the transport is passive transport. No energy is required. While if there is a need of energy expenditure and the movement occurs by the use of energy and against its concentration gradient, then that movement is known as active transport. Now again we can see here that diffusion means random molecular movement of substances molecules by molecule through intermolecular spaces. There is no energy expenditure by the cell, right? So if this is a lipid bilayer and these are the molecules, they diffuse into inside of the cell via passive transport or diffusion, right? So there is no protein required for the movement of these molecules, and diffusion generally occurs of those molecules which are soluble in lipid soluble. Only lipid soluble molecules can move through the cell membrane without the need of any carrier or channel protein for its movement, right? So if we move on to the next part, in contrast active transport means movement of ions or other substances across the membrane in combination of a carrier protein in such a way that the carrier protein causes the substance to move against the gradient. That is, the movement from a low concentration to high concentration state. This type of movement requires an additional source of energy, right? So as we already know that as seen in this picture as well, that in active transport here is a carrier protein required in the lipid bilayer, and although these molecules are in high concentration inside, still more substances have to be carried inside. That means the opposite to concentration gradient transport have to occur. Then active transport is used, where these protein carrier molecules bind to these molecules in its bases and then changes its conformation and releases this molecule on the other side of the membrane. Now diffusion through cell membranes can occur in two ways: simple diffusion and facilitated diffusion. Diffusion through cell membrane is divided into two subtypes: simple diffusion and facilitated diffusion. The simple diffusion means kinetic movement of molecules occur through intracellular spaces of the membrane without any interaction with the carrier protein, right? So as we can see in this case, the diffusion is occurring of lipid soluble molecules through the membrane without the need of any protein molecules, right? While in facilitated diffusion requires interaction of a carrier protein, the carrier protein aids passage of the molecular ions through the membrane by binding chemically with them and shuttling them through the membrane in this form. Like we can see in this cases, the molecules come through this protein molecule. It requires this protein molecule to pass through it, possibly because although the concentration gradient is high here and low here, still these are not soluble in lipids, so they cannot easily diffuse through the lipid bilayer. Hence they require a channel protein or a carrier protein. Now facilitated diffusion, as we already saw in this case, Facilitated diffusion requires interaction of a carrier protein. The carrier protein aids passage of the molecules or ions through the membrane by binding chemically with them and then shuttling them through the membrane in this form. Right? So it can be channel protein or carrier protein to which the molecules bind and then carried inside or outside of the cell depending on the concentration gradient. And facilitated diffusion, though no energy is required. No energy is required. Now, diffusion of lipid soluble substances through the lipid bilayer. Now, it is important to know that an important factor that determines how rapidly a substance diffuses through the lipid bilayer is the lipid solubility of the substance. For instance, the lipid solubility of oxygen, nitrogen, carbon dioxide is solubility of these substances is very high, and also of alcohols. So these substances can dissolve in the lipid bilayer and diffuse through the cell membrane in the manner that diffusion of water solutes occur in a watery solution. The rate of diffusion of each of the substances through the membrane is directly proportional to the lipid solubility. Especially large amount of oxygen can be transported in this way. Right? So we can say that oxygen, carbon dioxide are easily transportable because these are soluble in lipid molecules, while water requires protein called aquaporins to be transported from one side of the plasma membrane to the other, because water is immiscible in the lipid. Selective permeability of protein channels. Protein channels are highly selective for transport of one or more specific ions or molecules. Potassium channels allows the movement of only potassium ions, and sodium ions channel allows only the movement of sodium channels. Now if we see how that happens, so we can say that if it is a cation channel for a cation, then the channel will have amino acid present in them which will be negatively charged. Negatively charged amino acid will be present inside these channels if the channel is for a cation. So when negatively charged amino acid will be there, it will generally, in fact it would only attract the positive ions toward itself, right? Only positive ions will be allowed in it. While if it is a for an anion like chloride channel, then the amino acid lining the channel will be positively charged. Right? So that is that. But how can we differentiate between a potassium channel and the movement of sodium channel? Because both the channel will be a cationic channel and will have negatively charged amino acid. So for that we will say in the next slide. It is in picture of a potassium channel, and in this picture we can see that there is a selectivity filter, right? There is this selectivity filter through which only potassium ion can pass, right? So how this happens, this selectivity filter have a carbonyl oxygens, and when potassium hydrated ions comes into the selective filter, the water molecules are removed, reacting with this carbonyl oxygen, and only dehydrated potassium ions are allowed to pass through this selectivity filter, right? Only potassium hydrated ions will be allowed to pass through it, right? But if there is a sodium ion, then although the sodium ions are very small, and although they can enter this channel, but they could not be able to pass through because these carbonyls will be too far apart, right? So we will move for the next slide. Now we will study about the gating of protein channels. The gating of protein channels provides a means of controlling ion permeability of the channels. The opening or closing of two gates are controlled in two ways: voltage gating and ligand gating. Now the opening or closing of gates are controlled in two ways. In voltage gating, the case of voltage gating, the molecular conformation of gate or its chemical bond responds to electrical potential, as given in this example of sodium and potassium gates. So in this case, as we can see, the sodium gated ion channel is closed, while if a change in the membrane potential occurs, then the gate will be opened and sodium ions will be allowed to move in. While the same occurs in the case of potassium ions, where in the first case the gate is closed, while in the second case the gate is open because some potential change has occurred in the membrane, right? Some potential change has occurred in the membrane, so potassium will move out. So this is the example of two voltage gated ion channels. In this, we can see that voltage gating example, the molecular conformation of the gate or its chemical bond responds to electrical potential across the cell membrane. For example, when the membrane potential changes inside and becomes less negative, the sodium channel opens and allows sodium to pass inwards, while they close when inside of the membrane become very negative, right? So if the membrane potential changes and there is this positive outside and negative outside, so then the gate is closed. But if it changes a bit and it becomes less negative, then the sodium gated ion channel will move and sodium will move inside. While if the membrane potential inside becomes relatively positive, only then the potassium ion channel will open and potassium will be released outside. Potassium channels open when membrane potential becomes positive. Now ligand gating. Some protein channel gates are opened by binding of chemical substances, a ligand, with the protein, which causes a conformational change in the protein molecule that opens or closes the gate. Example, acetylcholine channel. So acetylcholine channel can be seen in neurons, right? If I make a neuron here, right? So this part will have many acetylcholine channels, like acetylcholine channels, and it would release acetylcholine into the synapse, and in the next cell will have a acetylcholine receptor, and when this acetylcholine will come and bind here into this acetylcholine receptor of the next membrane of the next cell, then the signal will be transmitted further. A gate will be opened, right? So this is the ligand gating, and in the next part we'll study about active transport of substances through the membrane. When a cell moves molecules or ions uphill against concentration gradient or electrical gradient using energy, the process is called active transport. Different substances that are actively transported through cell membranes include sodium, potassium, chloride, iodide, sugars, and amino acid, right? So those molecules which are lipid insoluble, and large molecules which could not be moved in, and the concentration gradient is, even though opposite, they are still needed to be pulled inside of the cell. The biggest example of this type is in every cell. The potassium ion is high inside every cell and sodium ion is low, while outside sodium ion is high and potassium ion is low. And this type of intracellular and extracellular is maintained, conditions are maintained, right? So maintain these conditions, active transport is required, which is done by sodium potassium pump. Sodium potassium pump, right? So this sodium potassium pump actively transports sodium outside the cell and potassium into the cell to maintain this condition. Now primary active transport, we already know that there are two types of active transport, primary and secondary. Active transport divided into two types, primary active transport and secondary active transport. In primary active transport, the energy is derived directly from breakdown of ATP. In secondary active transport, the energy is derived from the gradient set up by the primary transporter. In both instances, transport depends on carrier protein that penetrate through the cell membrane. Example of primary active transporter is sodium potassium pump that transports sodium into and out of the cell and potassium ion into the cell. Now sodium potassium pump. As we see in this case, the pump has two subunits, the smaller alpha and larger beta. This is alpha subunit and this is the larger beta subunit. It has 3 sodium binding sites on the portion of the sodium protein that protrudes inside the cell. Right, so this part has the binding site for 3 sodium ions. 3 sodium ions bind on the intracellular part, the inside of the cell. 3 sodium ions bind, while 2 potassium ions bind on outside of the cell here, and then they are transported inside and thus outside, thus maintaining higher potassium ion inside and higher sodium ion concentration outside. So as we can see, it does not occur along the concentration gradient, hence it requires ATP, right? It has 2 binding sites of potassium ion outside. The inside also have ATP binding site. The sodium potassium pump is important for controlling cell voltage. As we can see that the fact that sodium potassium pump moves 3 sodium ion to the exterior, and for every 2 potassium ion that moves to the interior of the cell means that a net 1 positive charge is moved to the interior of the cell, right? So there will be more positive inside. This action creates positivity outside of the cell but results in deficit of positive ions inside the cell. That is, it causes negativity on the inside. Therefore, the sodium potassium pump is said to be electrogenic because it creates an electrical potential across the cell membrane. Now, in secondary active transporter, we can see that when a large concentration of gradient of an ion had been created by the primary active transporter, then this energy stored as the concentration gradient can be used to transport other molecules along with that ion. This is known as secondary active transporter, right? So what it means is that, as we can see in primary active transporter, which is here, is the sodium potassium pump, carries 3 sodium outside and 2 potassium inside, right? So by this, a high sodium gradient, a high sodium concentration gradient will be created outside, and according to the law, it will always try to move inside of the cell because its concentration is higher outside. So this energy stored as the concentration gradient of high sodium concentration outside can be used to pull other material along with it, right? So here we can see a secondary active transporter like glucose sodium symporter, sodium glucose symporter. So this concentration gradient which has been created by the primary active transporter is used to carry a glucose molecule along with the sodium, right? So it does not directly use ATP, but it uses the energy that has been stored by the action of the primary active transporter. So the secondary active transporter is of 2 types, co-transport and counter-transport. Co-transport is when both molecules are transported in the same direction, as seen here in sodium glucose symporter. While counter-transport, or also known as anti-transport, is when 2 molecules are transported in opposite direction. As we can see in this case, although it is a primary transporter, Thank you for watching. If you want any written material, you can comment below.